Welcome to the 1-4 podcast, and this is on tools and procedures for studying biology. And the first thing we get to is the measurement system that most scientists use. And the measurement system they use is one that's common for everybody, so that scientists can replicate each other's experiments, they can communicate to each other about each other's experiments. It's like everybody talking the same language, and that language is the metric system. The metric system is based on a decimal system, meaning it's units of 10, and it's easy to use because everything's based on multiples of 10. You can convert from one unit to another just by multiplying or subtracting by a factor of 10 or 100 or 1,000, or divide by 10 or 100 or 1,000. And the system is called the revised system they use is called the International System of Units, or SI, which is French for System International. And here's the comic metric units that we'll use. Length is the meter, and you can see there are some other the other ones we might use might be centimeters, millimeters, which are 1,000th of a meter, and kilometer, which is 1,000 meters. And then for mass, the unit we use is the kilogram, which is 1,000 grams, and 1 gram and 1,000 milligrams is equal to 1 gram. The SI unit for volume is the liter, and you can see we have milliliters, which are 1,000 liters, and we also have cubic centimeters. Centimeters are cubed, which is basically the amount of volume that takes up one cube of centimeter by centimeter, and that's equal to 1 milliliter. Often when people talk in hospitals, I know my wife, who's a nurse, talks in terms of mils or cc's, mils being milliliters or cc's being cubic centimeters, and they're one and the same. And then the last unit that we'll spend a lot of time with is temperature, which we talk in degrees Celsius. And you can see freezing point of water is 0 degrees Celsius, and the boiling point of water is 100 degrees Celsius. For us, the freezing point of water is 32 degrees Fahrenheit, and the boiling point is 212 degrees Fahrenheit. And those are not very easy to remember. You just remember that water freezes at 0, boils at 100. Once again, unit of tens. Now, when that's really one of the big important parts of the chapter. This is about analyzing the data, and you've probably done this in your labs, but they collect data to find out whether certain factors change or remain the same, and often they will put the data in a table and then graph it in a graph. And of course, scientists use computers to gather, analyze, and present large quantities of data. And then you're allowed to. Then what you can do is use that data to make predictions about complex phenomena. Without computers, the genome, which is basically all the sequence of bases in DNA, could never have been decoded. And this I thought was interesting. This picture. This is a picture of what a home computer could look like in the year, I think that's 2006 or 2005. I thought that was very funny. Light microscopes and electron microscopes, and microscopes themselves are devices that produce magnified images of structures too small to see with the unaided eye. Light microscopes magnify images using visible light rays, whereas electron microscopes produce magnified images by focusing beams of electrons. You are going to use a light microscope in our classroom, and you've probably done so already this year. And they can produce clear images up to about 1,000 magnification of about 1,000 times. Our microscopes in our lab produce images up to 400 times, but you can get microscopes that can produce up to 1,000 times. And the compound microscope, as you're going to find allows light to go through the specimen. That's why the light is on the bottom of the stage. Two lenses, one being the objective, the other being the eyepiece. And you can and it allows us pretty good power to see even live animals when they're still alive. Now with an electron microscope, the limit is 0.2 micrometers, meaning 1/50th the diameter of a typical cell. But that's a really amazing clarity. There's a picture of one, and so an electron microscope allows us to see even smaller than that. And there, for instance, is the surface of some kind of spider or something. And electron microscopes use beams of electrons rather than light. And they can produce almost 1,000 times more detailed than light microscopes. So think about that, 1,000 times magnification for a light microscope, and then multiply that by another 1,000 times, and that's what an electron microscope can do for you. Now, cell cultures are a way of making reproducing material, and what you're doing is you're putting something that you want to grow into a dish with nutrient solution, and the cells then in there will reproduce, and this is what we call the cell culture. There's a picture of a cell culture. It's basically a dish with a gel in the bottom of it, and you can use these to test cell responses, study interaction between cells, select specific cells for further study, etc. But the cell culture is this dish with this nutrient on the bottom, which in this case is a pink gel there, and it allows the cells to grow and then reproduce, so you have more cells. Cell fractionation is a technique where a sample can be put into a test tube, spun around, and then it's separated into layers. For instance, here's a layer here, here's a layer here, and then here's another layer up here. So by spinning the cell around, you can separate the cell parts. That's a picture of they're put in a blender, then put into a plastic tube, and then they're put into what's called a centrifuge, and that's what this is. It spins around, and then what it does is it causes the cell parts to separate, with the most dense parts settling near the bottom of the tube. And then what they can do is you can take a pipette, micro pipette here, and pull parts of the layers out of the cells that you want to look at. You'll be doing this when you go to the DNA Learning Center. You'll be pulling parts of cells out, and you will take cells, you're going to put them in a blender, you're going to chop them up, you're going to put them in a centrifuge, you're going to use cell fractionation with the centrifuge, you're going to pull it out with a micro pipette. Working safely in laboratory, we're going to use safe procedures and we'll talk more about how to follow the safe practices. Most important, if you just read the rules and follow those rules, then you should be fine. Read the steps before you do the lab and all the safety precautions. Follow my instructions and the textbook directions exactly. Everything will be fine. And of course, when in doubt, ask me. Oh yeah, wash your hands, of course. Alright, and not only are you looking out for your own safety, you're also looking out for the safety of me and everybody else in the classroom, or any live animals that you handle in the lab. On to the quiz. And this allows them to replicate and also to communicate communicate their findings to each other. An electron microscope will allow you to see smaller objects with greater detail. The centrifuge is going to separate it by spinning. Cell cultures will allow you put the cell into the gel in the bottom of the petri dish, and you should be reading all your safety rules before beginning work. You should read the instructions for any lab before you even start it. And that's the end of this section.